Hello everyone. Now we are going to discuss about the cellular and molecular hallmarks of cancer. To understand cancer, how a cancer develop or a malignant condition develop, we need to understand certain mechanism or certain feature of cancer. This is a mnemonic I have made for myself. You can make according to yourself, as I believe mnemonics are very personal. SIA in Miami. So S stand for self-sufficiency in growth signal. Self-sufficiency in growth signals. Later we will discuss all of these hallmark one by one in detail. So first we just name all these 8 hallmarks. Self-sufficiency in growth signal. Second is insensitivity to growth inhibitory signals. So a cancer cell is insensitive to growth inhibitory signal. A stand for evasion of apoptosis. Normally cells, a mutated cell destroyed by apoptosis or eliminated by apoptosis, but in cancer, this cancer cell has developed ability to evade the mechanism of apoptosis and survive. M stand for altered cellular metabolism, or known as Warburg effect. We will discuss Warburg effect in detail later on. I stand for immortality or limitless replicative potential. Cancer cells are almost immortal. They divide limitlessly, and that's why they form a huge mass or tumor. A stand for sustained angiogenesis. As we know, for a new mass or growth, blood supply is very important for nutrient availability of nutrient or excretion of dangerous excretory material. This is very important for cell growth. M stand for metastasis and invasiveness. This is one of the most peculiar feature of a cancer, that it metastasizes from its site of origin to distant organ, or invasiveness to the surrounding tissue. I stand for ability to evade host immune system. Cancer cells have the ability to escape from the host immune system, and that's how they survive. Before going into the detail of all of these hallmarks, we need to understand certain mechanism or pathways. First is growth factor signaling pathways in cancer. How a growth factor affect the development of cancer and various pathways involved in it. A growth factor attached with growth factor receptor with the help of bridging protein activate the inactive RAS into active RAS. Active RAS then by two mechanism. One is RAF and PI3K will result into the cell proliferation by various mechanism. Like RAF further stimulate or activate MAP kinase, which ultimately result into the activation of transcription through MYC and D-cyclin. MYC on the one hand stimulate various pro-growth metabolism and increases protein synthesis, and cyclin D, as we know, this will help into the progression of cell cycle and result into the cell growth. PI3K stimulate AKT. AKT itself will result into the pro-growth metabolism stimulation, or AKT further stimulate mTOR, which increase the protein synthesis and ultimately result into the cell growth. This GAP prevents the hydrolysis of GTP and help in remaining RAS into the active phase. GAP stands for GTP activating proteins. PI3K stands for phosphoinositol 3 kinase, and MAPK stands for mitogenic activating protein kinase. PI3K is also inhibited by P10 protein that we will discuss later on. So this is a basic or prototype growth factor signaling pathway in cancer. First of all, a growth factor comes received by the growth factor receptor will result into the activation of RAS, and active RAS by various pathway will result into the cell growth. Now first very important thing in cancer hallmarks is self-sufficiency in growth signals. The cancer cells remain proliferating, or they keep on dividing even after the cessation of the growth factor. So first important topic we will discuss is oncogenes or proto-oncogenes. The proto-oncogenes or oncogenes code for oncoproteins, and oncoproteins are essential for the cell proliferation. Oncoproteins are not harmful as such, but on mutation they become harmful or may develop into a cancer. So there are 5 classes of oncogenes. They involve growth factors, growth factor receptors, protein involved in signal transduction, nuclear regulatory proteins, and cell cycle regulator. These 5 classes are included in oncogenes or proto-oncogenes. Now we will discuss them one by one. Growth factor receptors. Growth factor receptors, we will discuss these oncogenes on the basis of oncogenes, they are coded protein, and the cancer they are responsible for the development of. First is in growth factors is PDGF. In growth factor, first is PDGF beta, which encode PDGF beta protein, and this is associated with astrocytoma. Astrocytoma is a tumor of glial tissue. Second is fibroblast growth factor. This encode HST1 and FGF3. Mutation of these proto-oncogene will result into osteosarcoma, may develop in stomach cancer, bladder cancer, breast cancer, and melanoma. So this is the mnemonic I have made for this. You can form your own mnemonic. Another growth factor is TGF alpha, proto-oncogene involved in this TGFA. and mutation of this proto-oncogene is seen in astrocytoma. FGF, hepatocyte growth factor, FGF proto-oncogene. Associated cancers are hepatocellular carcinoma and thyroid cancer. The second category of proto-oncogene is growth factor receptors. EGFR family, in code for ERVB1, also known as EGFR, epithelial growth factor receptor. ERVB2, also known as HER2. ERVB1 mutation seen in adenocarcinoma lung, HER2 mutation seen in breast carcinoma and several other cancer. FMs like tyrosine kinase 3, in code FLT3 proto-oncogene, and mutation of this proto-oncogene is seen in various leukemias. A receptor for neurotropic factors, RET proto-oncogene, and mutation of this proto-oncogene will be seen in Mann syndrome A and B, familial medullary thyroid carcinoma. So medullary thyroid carcinoma, one of the most important mutation is seen in RET gene. PDGF receptor, in code for PDGFR beta, and mutation of this proto-oncogene seen in glioma and leukemias. Receptor for KIT ligand, mutation of KIT gene seen in GIST, gastrointestinal stromal tumor, seminomas and leukemias. ALK receptor, mutation of ALK gene seen in adenocarcinoma of lung, lymphomas and neuroblastoma. Now third category of proto-oncogene is proteins involved in signal transduction. First is GTP binding proteins. This involve KRAS, HRAS, NRAS, GNAC and GNAS. KRAS mutation seen in colon, pancreatic and lung tumor. Mutation seen in bladder and kidney tumors. NRAS mutation seen in melanomas, hematological malignancies. GNAC mutation will result into uveal melanoma. NRAS mutation seen in pituitary adenoma and other endocrine tumor. Non-receptor tyrosine kinase, proto-oncogene ABL. This will mutation of ABL gene seen in CML and ALL. RAS signal transduction pathway. VRAP mutation, VRAP gene mutation, and this is seen in melanomas, leukemias and colon carcinomas. Notch signal transduction pathway, in code Notch1 gene, mutation of Notch1 gene seen in leukemias, lymphomas and breast carcinoma. JAK-STAT signal transduction pathway, JAK2 gene mutation seen in myeloproliferative disorders and ALL. Fourth category of proto-oncogene is nuclear regulatory proteins. This include transcriptional activators like MYC and NMYC. Mutation of MYC is seen in Burkitt lymphoma and mutation of NMYC is seen in neuroblastoma. Fifth category of Protooncogenes, cell cycle regulator. First is cyclin, second is cyclin dependent kinases. Cyclin in cyclin, mutation of CCND1 or cyclin D1 mutation will result into the mantle cell lymphoma, multiple myeloma, breast and esophageal cancer. Cyclin dependent kinases CDK4 mutation will result into the glioblastoma, melanoma and sarcoma. Look, this is a very volatile topic, particularly including proto-oncogenes and tumor suppressor gene, but this is also one of the very important topic of pathology, and all of these genes are come again and again in front of you whenever we read pathology. So we need to mark it up this topic, keep revising again and again. There is no way to remember these volatile topic. The only way is to keep revising again and again. In the next lecture we will discuss about the tumor suppressor gene. So thank you.